Awesome. All right, so folks are wrapping up from other meetings. Why don't we get started, and folks will join as we go. I'm going to turn over to Mackenzie's going to welcome you all. But as folks are getting settled in, we're just going to start with some announcements. Welcome to our fall installation of the Knowledge Portal Network and webinar. And we have a guest speaker today, which we'll introduce in a moment, and some early opening comments. And we also will be adding to our webinar series in the fall and early winter, so look for some updates there. And I'm going to turn it over to Mackenzie for some announcements and just some housekeeping. Wenli is an incredibly flexible person, so she's going to be great to, if you have any questions as we go, just pop them in the chat or in the Q&A. You can even raise your hand if you feel like it. Again, these are, interactive and informational. And if you and just note these are being recorded, so if there's anything you like and you want to see later, it'll be on our YouTube channel for you to follow up on. And so with that, I'm going to turn it over to Mackenzie. He's going to walk us through some new and notables from the network before Wenli speaks.
Awesome. Thank you, Noel. All right, before we get started, I'd like to welcome a new member to our Knowledge Portal Network team, Alex Schillen. He has 20 years of expertise in web development, front-end design, and user experience. So he's joining us as a principal design engineer, and he'll be helping us with the new NIH-funded initiatives across the Common Fund and human adipose tissue. So just big welcome to Alex. Okay, so now I'm going to go through some of our upcoming events that we'd like to keep an eye out for. First off, we have our ancillary event at the ASHG conference. And so Noel and Jason and others will be speaking, and they'll be discussing the recent expansion work on human genetic evidence calculator and integrating other data sets and results to enhance gene prioritization. They'll also walk through some of our new production release items, which will be slated for the end of the month. So get ready for a new production release. And we'll also be exhibiting at the booth. So feel free to come visit our team. And if you're interested, you can receive a personalized demo. We'll be discussing the A2FKP, CMDKP, and CMDGA. So if you have anything that you'd like to go over with us, we'll be there. Okay, next up, we also have the new speakers for our upcoming dates for the Knowledge Portal Network meetings. So first up, we have Kyle, and then we'll have presentation from Anna Gloin. We'd also like to show here that we'll be adding a new date for November 30th for a focus group meeting. It's not on the calendars yet, but it's going to be added, and we'll be discussing new features. In addition, our 2024 schedule will be out soon, so please be on the lookout for that. And as Noel said, we'll be introducing a new webinar series for our new awards and projects. So we'll have a little bit more information for you coming soon. And so with that, I'd like to now introduce Dr. Wenli Yang, who is a research assistant professor of medicine at the University of Pennsylvania School of Medicine. She's also a member of the AMP-CMD consortium that's generating genetic and genomic results to accelerate the development of new therapeutics for common disease. In this webinar, she'll present her work on using stem cell-derived lineages for disease modeling.
All right, so today I'm going to talk about, as I mentioned, a brief view of the core that I run at Penn. And to start with this slide, I actually stole this from Paul Gaudu. I think Paul is on. Hi, Paul. I think Paul took it from a review. So this is just give sort of overview of what iPS cells can do. And I think a lot of you already know, but it's been a really powerful platform to study human disease. Obviously, you can identify patients that harbor various mutations and variants, and then you could isolate these cells, for example, blood cells, from the patients, and using those cells, you can derive induced pluripotent stem cells for iPS cell lines. And they're fairly straightforward to do. I'll go over a little bit about how we do that. And then using CRISPR-Cas9 or other methods such as TALEN, you can correct or introduce mutations in the iPS cell lines that you derive. And this is really readily doable as well. So once you introduce or correct the mutations, you can, using some really well-established methods, by many investigators in the stem cell field to differentiate cells into the cell types of interest. For example, neurons, beta cells, islets, and etc. So that you can, together with the control or isogenic control line, to study, to do disease modeling to look at gene function. You can use these derived cells to perform screens, such as genetic screens or small molecule screens to identify drugs that can reverse disease phenotypes or elucidate gene function. You can, of course, use the differentiated cells to assay gene function during development. So one really beautiful thing you can use these iPS cells for is you can look at a differentiation, different stages of differentiation from a stem cell to a differentiated cell type that you otherwise would not be able to look at in an animal system. So you can also use generate reporter cell lines. For example, you can put a tomato or GFP downstream of a gene, and therefore the cells will light up red or green, etc. And so they can mark the cell types you're generating. So to use to trace cells and to purify cells, of course, in order to enrich for the cell type of interest that you're interested in looking at. Of course, the ultimate goal is to use these cells for cell replacement therapy, and there's a lot of work in the clinical field that's happening right now. So this is sort of an overview of using iPS cells as a platform to study human disease and also for clinical applications. All right, so I'm trying to advance and I can't. Oh, here we go. So, I think I alluded to there's advantages of iPS cell and iPS-derived models. Of course, first is that these cells are renewable. They can be passaged indefinitely without, becoming senescent. So you can, you're not limited by the number of cells to work with. They're amenable to genetic manipulations. I mentioned about CRISPR-Cas9 gene editing. They can be differentiated into multiple cell types, of course. So you can, enabling analysis, apply tropic actions of the genes of interest. For example, the type 2 diabetes effector genes that I'm interested in. And I already mentioned it can be used to study developmental stage specific gene function in differentiating cells. So as I mentioned earlier, I run a stem cell core at the University of Pennsylvania, and as a core facility, we provide these services to the Penn and CHOP and the surrounding community. We do iPS derivation and characterization from somatic cell sources. I mentioned blood, but we can also use a number of other cell types, such as skin fibroblast, adipose stem cell, epithelial cells, for example, from urine, CRISPR-Cas9 gene editing to generate mutant cell lines. And I'll talk in a little bit more details for iPS differentiation. We can generate a variety of cell types in order to do disease modeling. We also train researchers who wanted to start using stem cells in their own laboratories. And we have quality test of stem cell culture media and reagents available, so you don't have to test yourself. And lastly, we have a large collection of normal control iPS lines, as well as some disease lines available to collaborators. So how do you make an iPS line from somatic cell? And here I'm just giving an example, making an iPS line from blood. And we've actually put this protocol in a STEM book, which everybody can access at stembook.org. So you don't need very much blood. So a couple mils of fresh blood, and from there you can isolate PBMCs. And from PBMC, which is a heterogeneous population of lymphocytes and other progenitors, you can expand out the erythroblast erythrocyte progenitors, and those are the cells that we then use to infect reprogramming or Yamanaka factors, which then can be the cells can be reprogrammed into iPSCs in 3 to 4 weeks. So this is just shows briefly how we isolate this, the PBMCs from blood, and what the cells look like, the erythroblast cells before we subject them to reprogramming. And over the course of 3 weeks, we can see the emergence of really nice iPS colonies on the bed of mouse embryonic fibroblast feeders. And then these are the cells that colonies are the ones that we pick, isolate, and further expand through serial passaging to really stably reprogram them into iPS lines. And along the way, we subclone them so that they're becoming clonal, followed by more enzymatic passaging and banking at various stages, so we don't lose the lines, and extensive characterization, including qPCR for stem cell markers, lineage-directed differentiation, in order to assay whether or not they're able to give rise to the three lineages: endoderm, ectoderm, and mesoderm. And we karyotype them, so we make sure they have not gained chromosomal abnormalities because of their programming process. And we compare their fingerprint to their somatic parental cell line cell source to make sure that these lines are indeed derived from what we think they come from. And again, and lastly, we subject them to pathogen testing to make sure they're free of various pathogens. So this process takes about 3 to 4 months. And then we bank them, and they can be used to differentiate at around passage 15 to 20 or so. I mentioned about extensive QC and characterization of the cells to make sure they're bona fide iPSCs and they're free of genetic additional genetic abnormality accumulating in these cells. And I think I mentioned earlier, we have quite a large collection of normal iPS lines in our lab, and this is due to a result of a large study that was funded by NHLBI several years ago, where we recruited healthy donors. Actually, they are 18 to 30-year-old, I believe. And we derived, more than 130 cell lines from distinct genetic background, and all the iPSC DNA have been fully genotyped, and 90 plus lines have been differentiated into hepatocytes and cardiomyocytes. So we know how they, whether they make endoderm and mesoderm well, at least hepatocytes and cardiac cells. We deposit all the lines at Y cell repository, but they're also available through collaboration if you want to use these for your studies. I should mention these lines really indeed provide a really rich source of common variants for investigations such as type 2 diabetes, SNPs that we're interested in. So we do quite a bit of CRISPR-Cas9 gene editing in the core lab, where this is a full service where we talk to investigators and understand exactly what they're looking for, whether they want to do a knock-in, a single base knock-in, or deleting a large chunk of DNA, or doing a reporter, a large knock-in, for example, putting a GFP reporter or a turbo ID for proximity labeling. Then we design CRISPR guides and validate them. And we also design and construct knock-in templates DNA sort of donor templates. We have pre-constructed reporter backbones available, so you can swap, things in and out easily. And then we obviously target the lines and screen and clones, and we deliver correctly targeted cell lines to investigator fully genotyped. And we have several sort of pre-validated control iPS lines for available for targeting. I believe we have 2 African Americans, several Caucasians, female and male as well. So this just goes through a sort of a workflow, which I think I just sort of mentioned for gene CRISPR targeting design. Order or make the guide RNA, validate in the iPSCs, make sure they efficiently cut at the site of interest, and then target and obviously verify the clones and give the cells back to the investigators. Here's just an example of a knock-in. We use a single-stranded oligonucleotide donor, for HDR. And on the right, it basically shows 2 clones. One is biolytic editing. So I should mention we engineer restriction sites here in the donor oligo that will give sort of a silent mutation, so without changing the coding sequence, and then we could assay for gain or loss of a restriction site when we do genotyping. So here, 2 bands means the restriction enzyme cut the sequence. So you have fully cut, you have a biolytic editing, you have a cut, and also you still have an uncut band here, which indicates a monolytic editing. Also quickly just reporter knock-ins. So we are involved in generating of a long alveolar type 1 cell SGDB3A2 cherry picker, a line that marks long epithelial cells, and when the cells are differentiated into long cells, they will light up red here really nicely in sort of organoid or spheroid, and growing in matrigel. And so this is sort of a summary of sort of gene editing efficiencies. So data gathered from targeting greater than 40 loci. So knockout is really efficient. Obviously, you're just making a cut and getting an indel formation. Deletion with 2 guides are less efficient, and knock-in without antibiotic solution is mostly inefficient, but it's definitely doable. So this can be done fairly straightforwardly, but requires a bit of work, and especially when you do single base knock-ins. So I want to spend some time talking about the cell types that we can generate. And here I'm showing you some of the cell types that we routinely generate, and this is not an exhaustive list. So that include skeletal myocytes or myotubes, iPS-derived hepatocyte-like cells, cardiomyocytes, adipocytes, retinal pigmented epithelial cells, and endothelial cells. So one of the first cell types that we're able to derive or differentiate are cardiomyocytes from iPSCs, and this is a fairly straightforward protocol, which is depicted here on the top. We make mesoderm, cardiac mesoderm, progenitors, and then beating cardiomyocytes, and we then subject them to the metabolic selection. So only the cardiomyocytes will live, and the non-cardiac cells will die off. And I'm sure some of you have seen this. Can you guys see? I'm not sure. The beating cells here. So you get a really nice plate of beating cardiomyocytes plate, and they are over 90% cardiac troponin TNNT2 positive, so fairly pure. And when you stain them for cardiac markers, and they stain really nicely. And Kira Mishinuro's lab, Natalia Petrenko, did some patch clamping for us. And if you compare ventricular adult human cardiomyocyte to our iPS-derived cardiomyocytes look sort of similar. And if we treat the cells with isoproterenol, which activate beta-adrenergic signaling receptors, you can see the cells beat faster than the cells that is not treated with isoproterenol. Here, I think, this is done in collaboration with Ben Prosser's lab, where he infected cells with adeno a beta tubulin, which lights up the cells, and you can see the in a subcellular level, the cells are contracting, and this is on the bottom. It shows one cardiomyocyte on a beta matrigel contracting really nicely. So during COVID, we actually collaborated with Susan Weiss's lab, who's an expert at Penn on coronaviruses, and we generated iPS-derived cardiomyocytes and give it to her lab, where she is able to infect these cells with SARS-CoV-2 really, really nicely. As you can see in panel B, here you can see that cells can be infected with SARS-CoV-2, which is stained really nicely in green with the antibody against a protein expressed by the virus, and then the cardiac troponin T antibody staining for cardiomyocytes. Her lab has gone on to show that the cells SARS-CoV-2 replicates when infected after infecting iPS cardiomyocytes, and induces innate immune response in these cells. So subsequently, we actually made it using CRISPR-Cas9 and ACE2, which is the receptor by which the virus gets in an iPS line, and when we differentiated the cells into cardiomyocytes, we saw loss of ACE2 expression in the cells. So this would be a really nice tool for people who are interested in using these iPS-derived cardiomyocytes to look at SARS-CoV-2 pathogenesis. Another cell type we actually derive and actually propagate in the lab are long alveolar type 2 cells. These is another cell type that's one of the major cell types that's infected by SARS-CoV-2, and these cells secrete surfactant proteins to help with surface tension when breathing. So this is a protocol that's being developed and published by Daryl Cotton's lab at Boston University. It takes 30 days, and then you sort the cells. Well, I should say the surfactant protein C is marked by a TD tomato. So this is using CRISPR-Cas9 to generate a reporter, and that's why the cells, once differentiated into the IAT2 cells, look red. So once these cells are differentiated, they can be propagated. So you can singularize the cells and reseed them in matrigel, and they can reform these spheres, these organoids, and you can serially passage them every 2 weeks, and they will regrow and redifferentiate. So we generated these IAT2 organoids and give it to Susan's lab, where they were able to use the SARS-CoV-2 virus to infect these cells. Again, on the bottom right, you can see, similar to the cardiomyocytes, they're readily infectable by the virus, and then elicit a similar innate immune response, as I just showed you in the previous slide. So another cell type that we actually been differentiating for some time, and can really easily differentiate or generate from iPSCs are hepatocyte-like cells. And the reason they're called HLCs because they, by the field, is because they are not like mature hepatocytes in that they still express quite a bit of alpha fetoprotein, which are expressed obviously by fetal hepatocytes, not mature hepatocytes. So this is a protocol schematic shown on the left. It's takes about 21 days or 3 weeks to do. And at the end, you can see the transcription factor, HNF4 alpha, stains really nicely in the HLCs, and a surface receptor, ASGPR1, is also nicely lights up when we stain these cells with antibody against the protein. So I mentioned earlier that we were able to generate HLCs from 90 plus iPSCs generated from normal donors. We did RNA-seq on all of the HLCs, compare them to human primary hepatocytes in peach here, and as well as liver samples. The gene expression level, as you can see, they're not quite on top, or they're not on top at all compare of the hepatocytes with the liver, but they're also different from the iPSCs. So they're somewhere in between, and therefore they're called HLCs. However, they express a lot of the genes that the hepatocytes express, and they're sort of is good enough to use as a as a model for a variety of disease modeling, which I'll show you in the next slide. So we use these HLCs to model A-beta lipoproteinemia. So, and this has been published several years ago. This is done in collaboration with Dan Rader's group and Ed Morrissey's group. So ABL is a rare genetic disorder, and it's caused by loss of function mutation in the ER resident protein, MTTP, a microsomal triglyceride transfer protein. So this protein promotes lipid transfer to apolipoprotein B that's within the ER, therefore facilitating its secretion by the liver cells. So in MTTP deficiency, you will have a depletion of ApoB lipoprotein in the plasma, so it's unable to be secreted. While it's known that ABL patients exhibit hepatic steatosis, primary hepatocytes from patients had not been characterized. So we generated iPSCs from an ABL patient that has a MTTP mutation. It's a loss of function mutation, we think, and we derived iPSCs, and we also corrected mutation to wild type using CRISPR-Cas9. So on the next slide, you can see the left is sort of the correction using CRISPR-Cas9. On the right, so we then differentiated these cells, control and disease and corrected iPSCs into HLCs. As you can see, there's less apolipoprotein cellular ApoB in the MTP mutant HLCs, but in the corrected HLCs, you have a restoration of cellular ApoB levels. If you look at secretion here, you see almost no secretion of ApoB into the medium, but a restoration of secretion when we corrected the mutation. And again, on the bottom here in D, you see accumulation of fatty acids, so which indicates sort of steatosis in the mutant HLCs, as shown with royal red O staining, and the corrected line had sort of a restoration of the defect. So I just want to quickly go through another cell type that I mentioned that we generate in the core, which is retinal pigmented epithelial cells. So RPEs is a pigmented cell layer that maintains photoreceptor integrity. And it's firmly attached to the underlying choroids, which is the microvasculature in the eye. And this is separated by a basement membrane barrier called Brooks membrane, here as depicted on the left. So RPE function includes phagocytosis of retina waste, immune modulation, light absorption, epithelial transport, etc. is quite important, a layer of cells. So in age-related macular degeneration, which is also called AMD, so this disease is a progressive degeneration of the RPE layer and the overlaying photoreceptors as well. So eventually leading to loss of central vision. So when you look at someone, you can see sort of a hole in the middle of their face in people with advanced AMD. So a hallmark of AMD is increased drusen formation, which is these extracellular fatty acid fatty protein deposits that contain apolipoproteins or mainly ApoE between the RPE and Brooks membrane. So obviously the genetic factors that contribute to pathogenesis of AMD. So we teamed up with Dan Haas group, who's a faculty at Penn in the Department of Bioengineering to model AMD using sort of a tissue or RPE choroid complex on a chip. So they're really good at making microfluidic devices that you can then seed cells into devices, so you can layer the cells. You can contain different layers, different, in this case, the RP layer and the Brooks, the microvasculature in different areas of a device, and so that you can overlay cells on top of each other. So we team up with them to see we can come up with a model for AMD. So first we took a protocol to make RPEs from Sally Temple's lab, and we were able to show that we can derive iPSC RPEs fairly nicely. Here, you can see RPE cells derived from iPSCs show pigmented patches, and then we can isolate these patches and propagate them to get homogeneous layers, populations of RPE cells. And here on the C, you show gene expression of 2 wild-type iPS line derived RPEs, and they express no markers of pluripotent stem cells, but markers of RPE cells. And these RPE cells form tight junctions as evidenced by ZO1 and staining, and they can do phagocytosis as evidenced by uptake of these pH-sensitive particles here. And as compared to primary RPEs, and they morphologically look really similar, and they're able to form sort of drusen as evidenced by accumulation of ApoE formation, as you've seen by the yellow staining here. I won't go through here, but on the right, but we concluded the iPS RPEs behave similarly to primary RPEs. And then we further, so we obtained iPS lines from AMD patients and differentiate these cells into RPE cells. And we can show that there's similar differentiation between normal and AMD RPEs at the gene expression level, and also morphologically on the right. But interestingly, when we looked at drusen formation, compare between control and DMD sort of RPEs or in the microfluidic devices the dance lab has engineered, and in which we overlaid these cell layers, and we saw there was increased drusen-like deposit in the AMD patient RPs as compared to the controls, and also the phagocytic function of the AMD RP cells also decreased as compared to controls. So it looks like the device or the dance lab has engineered is working, and then the next step is to use it to as a model to look at AMD. So I should mention the before I showed that the microvasculature here, obviously you have to form in the device, use endothelial cells and fibroblasts to form to generate vessel cells. So the endothelial cells used here are HUVAC cells that are purchased, and then the fibroblasts also are purchased as well. So the next step we wanted to do is using iPS-derived fibroblasts as well as iPS-derived endothelial cells to reconstitute this microfluidic device. So I think that's something that we're interested in doing, collaborating with Dan's lab for the next step. So the last few minutes, I want to switch gears and talk about actually something that that's so why that's one of the reasons why I'm giving a talk here with the portal webinar is because I'm involved with AMCND in this large sort of grant, which is functional interrogation of T2D associate genes using stem cells and mice. So here is a Penn group. It consists of computation genetics folks, genomics network analysis folks here, and as well as human cell and animal based genetic analysis group on the right here. So in a nutshell, so what we wanted to do is so working with the functional so genetics group to mine or narrow down a list of T2D associated genes, and that we can further use our models or tools such as iPS-derived models, primary cell models, and mice, and to look at function of these genes. So to that end, sort of part of my job is to come up with these iPS-derived model system to look at gene function. My group has also an interest in looking at T2D effector transcripts in a contactive skeletal muscle, but I won't talk about that today. So to that end of coming up of optimizing IPS models, we wanted to set up, at least able to differentiate or generate iPS-derived cells, metabolically active cells, such as adipocytes, muscle cells, livers, and islets or pancreatic islet cells. So, so I think I've shown this before to the AMP-AMD folks. So we're able to, using established protocol, we put a few tweaks in this protocol to make to generate iPS adipocytes. So here on the top right is just a schematic of how we make these cells. So it takes quite a long time. We generate adipogenic precursors, which we can freeze them down, and then you can thaw them later, and then induce them to differentiate into adipocytes. So on the bottom, you can see stained with Bota P here for neutral lipids, you have a really, really nice differentiation into adipocytes compared to the primary human adipocyte generated with progenitor cells that are isolated from biopsies. They're pretty, really, really nice. Interestingly, or not really interestingly, something that's nice for us to see is that these cells responds really nice to insulin stimulation for glucose uptake. Here's the dose response curve from no insulin all the way up to 100 nanomolar insulin. These adipocytes take up glucose really nicely, and it's really comparable to the primary human adipocytes. So we can use these cells to as a model to look at gene function in the adipocytes and generate mutations and reporters and etc., as I talked about before. Another a system that we developed is iPS-derived skeletal myotube cell myocytes. We combine a couple of protocols to make a myoblast, which again, similar to the fat cells, we can freeze and thaw and then induce them to differentiate or to fuse into myotubes. On the bottom, you can see really nice myosin heavy chain staining of myotubes 4-day post differentiation from the myoblast. And on the right, it just a qPCR showing transcription factors, structure proteins, and fiber type genes are appropriately expressed in this model. And we did some RNA-seq of these cells. So they are quite similar to human primary and immortalized muscle cells. Here, the immortalized muscle cells is the M2 line that's immortalized from a human myoblast. And then we also purchased primary human myoblast and differentiating into myotubes. But the problem is, I think I talked to this before in the sort of the MCMD group, is that these cells do not respond to insulin in terms of taking up glucose. They respond to insulin signaling peripherally, so as assayed by AKT phosphorylation. So on the bottom, I show iPS-derived myocytes. On the top, it's the M2 immortalized cell line. AKT is phosphorylated upon stimulation with insulin. However, as shown on the bottom here, the iPS-derived myotubes do not take up glucose in an insulin-responsive manner as compared to the M2 cells, and also compared to the primary human myotube. We tried a number of things. We have not been successful to get them to be responsive. We tried GLUT4 overexpression via lentiviral transduction, and we have somewhat inconsistent results in terms of glucose insulin responsiveness for glucose uptake. But these myotubes, however, have strong mitochondrial function. So in terms of assay by respiration, here's a seahorse assay. So we could use these cells for sort of to assay for mitochondrial function, of course. So just to summarize, where we are here. So I think I mentioned the iPS-derived models are the advantages that they can be introduced. They can be used to introduce variants for highly prioritized targets or genes that you're interested in to study mechanisms of gene regulation. However, some differentiated cell types, and I give an example of myotubes and also hepatocytes, which I didn't mention, do not functionally respond to insulin. In terms of using them for functional assays, such as glucose uptake and glucose output for hepatocytes. So for muscle cells, we have several things ongoing, which I don't have data to show you as yet, is that we're trying to co-culture with motor neurons to see if that stimulation will increase our insulin responsiveness. We also are trying 3D differentiation in different ECM with Chris Madel here at Penn to see if that would increase their insulin responsiveness. And of course, it's something that the several labs and folks that have tried is changing culture conditions for these cells, such as reducing insulin and glucose levels in both culture conditions and also differentiation conditions to see if they are more sensitizing them to insulin, because obviously the insulin, the glucose levels in these cells are extremely high. If we're using normal protocols. So the last couple minutes, I want to just talk about one piece of data that we have really recently obtained, and this is a project that we were funded from MCND, the opportunity pool. And the motivation for this project is that there is a lack of a set of well-characterized pluripotent either ES or IPS lines that can be shared across our consortium for I guess number one, robust and reproducible differentiation into multiple metabolic cell types that are relevant to T2D, such as hepatocytes, adipocytes, and muscle, etc. And also we lack lines are tunable and that are amenable for tunable and reversible perturbation of candidate genes for mechanistic studies. What I mean by that is that we don't have sort of inducible system where we can differentiate the cells first into, for example, hepatocytes, and then turn the gene on and off. We can turn the gene off permanently in iPSCs and differentiate them. But what if the genes is important for differentiation? Then we will never be able to assay that in a differentiated cell type, because they won't be able to differentiate right into the cell type of interest. So that obviously there is existing inducible IPS CRISPR I and CRISPR A systems being published. So I give an example here. One example published in 2016, where they engineered a transgene in the AAVS1 locus. This is considered safe harbor locus. This is DCAS9 crab. So this is CRISPR I. And has an mCherry sort of a marker just to see what the cells are being integrated with this transgene. However, the cells are, if you add docs to the iPS cells, the Cas9, DCAS9 is inducible, but you're unable to induce it further in differentiated cell types. For example, cardiomyocyte. So that means the locus gets silenced when iPS cells are differentiated. So that's no good, right? So if we, if we differentiate these cells into cardiomyocytes or hepatocytes, etc., we want the DCAS9 still be tunable and be able to upregulate it upon docs addition. So we turn to another locus, as was published several years ago, which is the Clibo locus. This is also safe harbor locus, similar to the AAVS1, but it has been reported that this locus has been shown to have improved transgene expression, higher transgene expression, and also is sustained transgene expression as compared to AAVS1 locuses. So implicating that this locus might be more open, that it's less prone to be silenced when the cells are differentiated. So similar approach, except it's a same construct. I forgot to put the mCherry marker here, but integrate into the Clibo locus. It took us some time to get this going because the the transgene is really, really large. So finally, we have 3 clones, which I've shown you here, 2B4, 2D4, and 2D5. In iPS cells, it's inducible. So it's a Western blot for dCas9, which is tagged by an HA. So it's a Western against HA epitope. All 3 lines are inducible, except 2D5, it seems it's less inducible here. So we went ahead and differentiated the cells into hepatocytes, and this is just got this a couple days ago. And this is still in the process differentiation, I believe stage 10 immature HLCs. And you can see that the mCherry still marker is still on. That means the transgene is active. So we are going to differentiate these cells in 3-week course and look at by Western blot for Cas9 expression, and to see whether or not the dCas9 can still be induced in differentiated cells. So this is endoderm. Obviously, we're going to do cardiomyocytes and adipocytes, which are mesoderm and muscle cells as well, and probably not probably, and also induce them to differentiate into beta cells. Of course, that's of a lot of interest to the consortium. So I think I will stop here and acknowledge my lab, which is shown on the bottom. Everyone in the group has contributed to what I talked about, and as well as the AMP, the common metabolic disease collaborators, and we have also the project for type 1 diabetes, which I didn't mention today. And of course, our collaborators over the years that we worked with in the middle. Thanks very much. I think there's a couple minutes for questions if anyone has any. I'll stop here.
Thank you, Wenli. Does anyone have any questions?
No questions.
Okay. All right. Well, thank you so much for taking the time to present today. And we look forward to hosting everyone in a month or so. All right. Thank you so much.
Great. Thank you.
Bye.